Good evening, everyone. I'm Aparna, a second-year PhD student, co-supervised by Professor Shomnath Bakshi and Professor Diana Fusco. And I've changed the title of my talk slightly. Can phages infect and eliminate dormant antibiotic-tolerant bacteria? Now, as can be seen from the schematic, phages being obligate parasites rely on the host cell machinery to make copies of themselves. And so if the host is dormant, the kinetics of the steps in infection are going to be impacted. And majority of the microbes in nature are in a dormant state. How does that impact their interaction with phage? And why is this an important research question? Phage therapy has stepped in as a potential solution in the fight against AMR, but despite the possible futures, there are obstacles that remain to be talented. Now, when we talk about antibiotic tolerance and persistence, when you treat an isogenic population of bacterial cells with a drug, the rapidly dividing bacteria get targeted and killed, but the slow-growing bacteria persist through the treatment, and once the drug is removed, they can restart growth and lead to chronic infections. So the question is, can phages infect and eliminate dormant pathogenic bacteria? Now, we perform high-throughput single-cell analysis of cell-to-cell heterogeneity using a microfluidic device, namely the mother machine, which contains individual lanes that comprise of trenches that allow you to load cells in single file and track more than 100,000 individual cell lineages under spatially uniform growth conditions. We treat cells with antibiotics and phages and study single-cell response over time. Here, we have treated dormant E. coli with ampicillin in depleted media for 7 hours, during which time you can see that the cells neither grow nor do they lyse, after which the cells are allowed to recover with fresh media, and it can be seen that the cells exit dormancy, elongate, and continue to divide. So dormant cells, as can be seen, survive ampicillin treatment and restart growth. Can phages infect and eliminate dormant antibiotic-tolerant bacteria? The single-cell phage infection assay that has been established in our lab by a senior PhD student relies on a slightly modified mother machine design, which comprises of these side trenches that allow phage diffusion and absorption onto the mother cell. This is an example trench in which cells have been lined up and the phages are allowed to flow past and infect the cell. And the phage infection assay that has been established includes staining the T7 DNA with a non-specific dye, namely Cytox orange, and the phage has been engineered to express a YFP reporter driven by the phytin promoter. And in this time lapse, the bright spot that you see here indicates a successful phage absorption event. And if you track the fate of the cell, it lights up showing successful infection and then proceeds to lyse. This assay has been established on exponentially growing E. coli cells, but can phages infect dormant E. coli? As part of the experimental setup, we inoculate E. coli and incubate it for 6 hours to allow the onset of stationary phase. We then further incubate the culture for 12 hours to generate a dormant culture that we spin down to generate the depleted media. And the dormant cells are loaded into the mother machine. The depleted media is spiked with T7 and flown in. And we collect time-lapse images, and namely 2 pieces of information are collected. One is the bright field image, and 2 is the YFP expression, which tells you about the stress response activity of the cell. So if the cells light up, it means that they are under stress. And here, the gray curves show you what the cell length looks like when the cells exit dormancy and enter exponential phase, where they grow exponentially and divide. In comparison, by maintaining an aeration ratio of 1:10 of culture volume to flask volume, we have ensured that the cells exhaust all nutrients and enter a state of deep dormancy, as can be seen by the pink traces, where the cell length does not change. And in the first experiment here, in the top panel, you see the no treatment control, where you treat dormant cells with spent media for 7 hours, during which time nothing happens, after which you allow the cells to recover with fresh media. They exit dormancy, elongate, and divide, and there's a reduction in the stress response activity. As you can see, they become darker in appearance. In the ampicillin treatment, you treat the dormant cells with ampicillin in depleted media. The cells don't lyse, nor do they divide. After that, you recover with fresh media. The cells exit dormancy and proceed to divide. Now, if you quantify the population demographics, In the no treatment control, 75% of the cells exit dormancy during recovery. And this percentage remains more or less the same, exactly the same actually, in the ampicillin treatment experiment, showing that dormant cells survive ampicillin treatment and restart growth. In the phage infection experiment, we treat cells with T7, infect them with T7 in depleted media. You can see that these cells proceed to lyse even under dormancy conditions, and even the cells that manage to remain unlysed do not show any signs of growth. And this is the no treatment control. And in the T7 infection experiment, we see that any cell that had the potential to exit dormancy get lysed by T7. So does a tendency to exit dormancy predict phage susceptibility? And can we predict a cell's potential to exit dormancy from the pre-exit cell physiology? Here, what you're seeing is the cell width of cells that exit from dormancy in the no treatment experiment. And it appears to be significantly lower as compared to cells that remain dormant. And this trend holds in the T7 infection experiment, where the cells that end up being lysed tend to have a significantly lower cell width as compared to cells that remain unlysed. If we look at the cell length, the cells that exit dormancy tend to have a higher cell length as compared to the ones that stay dormant. And the trend holds in the T7 infection experiment, where the cells that end up being lysed tend to have a higher cell length. So the next questions that remain to be answered is, can we track the steps of T7 infection progression in these dormant cells? And is there a correlation between the cell physiology and the lack of infection progression in cells that remain unlysed? To answer these questions, we have developed 3 reporters to track the progress of T7 infection steps in dormant cells. The first reporter tracks a successful adsorption event of a phage onto the cell. The second one tracks partial genome injection and T7 RNAP expression. And the third of complete genome injection and T7 capsid expression. And in this chymograph, you see that some cells just before lysis light up blue, indicating successful T7 RNAP expression, and some cells also light up yellow, indicating successful T7 capsid expression. And next, we aim to quantify the expression of these reporters and correlate how far the infection proceeds in unlysed cells. And I hope to come back and share the updates with you. Would like to thank my supervisor, without whom this talk would be impossible. I mean, most of the experiment designs and sketches were conceived by him. I happen to have the good fortune to execute them and to come to you today and talk about them. This is my co-supervisor, Diana Fusco. She is the brains behind all the phage experiments. And these are my labs. That's Bakshi lab, and that's Fusco lab. I would like to thank my lab members as well. Thank you.